In question 61, we're given this diagram here, and we're told that it represents the apparatus used by a student to carry out an investigation into the rate of photosynthesis in pondweed. So we have our Canadian pondweed here, and we have it producing gas bubbles when exposed to light. Now we should know that light engenders a photosynthetic process in plants, which generates gaseous oxygen, which is then used by the plant for respiration. But in this case, some of the oxygen is disappearing into the water, and then it passes along this capillary tube, and we can measure the movement of these gas bubbles along a capillary tube using this scale. Okay, so given that, we can say that, given that the rate of photosynthesis, or rather that photosynthesis is an enzymatic process, we can assess the rate of the enzymatic process of photosynthesis by the distance moved by the gas bubble. Okay, we're told that the student then calculates the volume of gas produced using the distance moved along by this gas bubble, and we're told that the diameter is 1 millimeter and the distance moved in 3 minutes was 16 millimeters. Okay, so the two things we need to work out for the answer is, firstly, the volume of gas produced, which we can work out using the movement by the capillary tube alongside the radius, which we'll come to in a moment, and also, given that we're told that there is a decrease in the rate of gas production after 3 minutes, what could be the reason for this? So that's the second thing we need to work out. Well, let's start by doing the mathematical part of the question, which is the volume of gas produced given a certain movement along the capillary tube. Well, how do we do this? Well, we have to model this as a cylinder, which we should know the equation being pi r squared h, with our h being the distance along the cylinder, i.e. the height, and the radius being half the diameter, which we're told is 1 millimeter. And we multiply that by pi. So we have pi times 0.5, our radius squared, times our distance moved, or height, which is 16 millimeters. Make sure the units are all the same, which they are in this question. So we have pi times 0.25 times 16, which is 1 over 4 pi times 16, which we should know is 4 pi. So we have our answer for the volume of gas produced, which is equivalent to 4 pi in millimeters cubed. Now, given that there are a range of different possible answers given for the reason that gas production would be reduced after 3 minutes, we can use the fact that we already know our answer to the left-hand column being 4 pi to whittle down the number of potential reasons. Well, let's have a look at the two which correspond to this answer. Firstly, all the enzyme active sites are occupied. Secondly, the carbon dioxide concentration is too low in our test tube. Now, which of these could be a reason for a reduction in the rate of gas production after 3 minutes? Let's think about this. So we have a reduction in the rate of photosynthesis. If we were to assess our first option, i.e. that all the enzyme active sites are occupied, well, we do know that as we increase the concentration of enzyme, after a point, the rates will eventually plateau, as, assuming substrate is the limiting factor in the transition from enzyme and substrate to enzyme-substrate complex to enzyme and products, changing the concentration of enzyme will eventually have no effect on this process. So it will eventually plateau, but the rate never decreases, so we can bear that in mind. The second option, the carbon dioxide concentration is too slow. Well, we know that carbon dioxide is the substrate in photosynthesis, as carbon dioxide and water are used to generate oxygen and glucose in photosynthesis. So, what we're talking about here is a lowering of the substrate concentration. Now, we should know that as substrate concentration decreases, the rate of photosynthesis decreases, as the rate depends on the transition of the substrate to an enzyme-substrate complex and eventually the products. So, the lowering of CO2 in concentration over the period would lead to a decrease in the rate of photosynthesis. So we can say that since the rate doesn't decrease for option A, or option 1 rather, we can say that this is wrong, and carbon dioxide concentration, i.e. substrate concentration, is the limiting variable here. So we finally have approached our answer. Let's quickly go through the final conclusion, which we know to be wrong since it corresponds to an incorrect value for the volume of gas produced. Let's just talk through it anyway. Photosynthesis enzymes are denatured. Well, we know there's no evidence to suggest that this would happen over the course of 3 minutes, as there is nothing affecting the efficacy of the enzymes at 3 minutes relative to the start, so we can say this is wrong. And we have our answer with 4 pi and carbon dioxide concentration being too low, and that's B. So that's the answer to the question.